My poster looks at computational analysis of transcriptomic data in PTEN-loss prostate cancer samples to identify neoantigens from alternative splicing events. Neoantigens are a type of tumour-specific antigen that can arise from disruptions at the DNA or RNA level. The tumour mutational burden is defined as the overall number of mutations in the coding region of the genome, which varies across different cancer types. Theoretically, it would be expected that the higher the tumour mutational burden, the increased genomic instability, and thus an increased number of neoantigens generated. Consistently, many studies have found a positive correlation between tumour mutational burden and response to immune checkpoint blockades, and limited efficacy of immune checkpoint blockades in cancers with low tumour mutational burden, for example prostate cancer. This suggests investigating neoantigens from non-mutational sources, which could help better understand the mechanisms of treatment response and resistance to immune checkpoint blockades, which is especially important in cancers that lack high tumour mutational burden. PTEN is an important tumour suppressor gene that is involved in many biological processes. However, a newer role for nuclear PTEN has been described where it interacts with spliceosomal proteins and drives pre-mRNA splicing through a phosphatase-independent manner. Alternative splicing is an important mechanism of gene regulation, and it contributes to transcriptomic and functional proteomic diversity. Most importantly, alternative splicing has been identified as a potential source of immunogenic neoantigens in low tumour mutational burden cancers. PTEN-loss has been correlated to an immunosuppressive tumour microenvironment. Neoantigens arising from PTEN-loss-associated alternative splicing could be relevant to the altered immune tumour microenvironment observed in PTEN-loss samples. The schematic on the right demonstrates the hypothetical impact of PTEN-loss-associated alternative splicing on the tumour microenvironment. Briefly, we suspect brown alternative splicing as a result of PTEN-loss can result in the formation of neoantigens. These neoantigens will go through the antigen processing and presentation mechanism, which would lead to neoantigens loaded onto MHC molecules and displayed on the cell surface. These neoantigens could then have potential inhibitory or promoting effects on immune cells. Overall, our work is novel and looks to identify PTEN-loss-associated AS events and predict alternative splicing-associated neoantigens. The first part of my research in my PhD focused on establishing a method to identify alternative splicing events from RNA-seq data and then predicting neoantigens from these events. Most algorithms available predicted neoantigens from mutational events, and to date, there are limited pipelines available to do this from alternative splicing events. From the published methods available, I have adapted these to use the latest machine learning neoantigen prediction algorithms to filter out a list of putative AS-derived neoantigens under PTEN-loss conditions. This has been first applied to D145 cell line data, which have been treated with SIP10, where peptides are filtered based on presence in the SIP10 group and lack of in the matched NSIP10 group. Importantly, as both these cell lines are cancer-based, the results from the cell line data work are only PTEN-loss specific and not cancer specific. However, later employing the TCGA data will allow us to identify both PTEN-loss and cancer-specific neoantigens. HLA typing is performed so that the putative neoantigens are sample-specific. The peptides are translated from the alternative splicing events and filtered to exclude those found in the reference peptidome, and then filtered further on percentile rank of the binding affinity of that peptide. Finally, we integrate matched mass spectrometry data to confirm the translation of these peptides at the proteomic level. To identify neoantigens, machine learning neoantigen prediction algorithms need to be employed. These algorithms have been trained on data from different points of the antigen processing and presentation mechanism. As shown in this figure, there are several steps to this mechanism, and training datasets either implicitly or explicitly represent the peptides from each stage. The most popular algorithms can be regarded as those that fall under the NetMHC banner, which have been updated several times over the last decade to extend the training datasets used. My research looks to identify strong, computationally predicted neoantigens, and so I'm exploring applying different machine learning algorithms to my datasets. So far, different machine learning prediction algorithms have been applied to D145 cell line samples that have been treated with SIP10, and a filtered list of putative in silico predicted neoantigens have been created. Matched mass spectrometry data has also been processed to obtain proteomic validated neoantigens from this dataset. These same pipelines will be applied to TCGA prostate cancer data. PTEN-loss has been correlated to an immunosuppressive phenotype, and therefore employing clinical data will allow us to investigate this by applying gene set enrichment analysis methods to PTEN-loss stratified data. Overall, this project will integrate RNA-seq and mass spectrometry data from cell line and clinical samples to characterize the role of PTEN-loss on alternative splicing and to identify whether alternative splicing-derived neoantigens are more suitable to investigate in low tumour mutational burden cancers. I hope this talk has highlighted the rationale behind my PhD research and the importance of computational tools in predicting alternative splicing-derived neoantigens.